This section consists of 3 introductory lectures. The first is an introduction to how we learn about and how we study about and how we understand metabolic pathways. It gives us a framework for how to learn about and study the metabolic pathways that we're going to discuss in biochemistry. The second section is an introduction to the regulation of metabolism and some general concepts about how metabolism is regulated. The third is an introduction to carbohydrates, their basic structure and function. So first let's consider how we learn about metabolic pathways. Now this first slide is a schematic diagram of all of metabolism, including carbohydrates, fatty acids, other lipids, proteins, amino acids, nucleic acids, and so forth. It looks pretty complicated, and in fact it is. And to many students this is a very daunting task to try to learn all of these metabolic pathways and how they're integrated together and this very complex system of regulation that occurs in every one of our cells. We want to, the lectures in this biochemistry series deal with the various metabolic pathways and discuss a lot of details about enzymes and products and substrates and so forth. But memorizing all the small details of these pathways is not a very efficient way to study for the boards. Instead of memorizing each and every small step, it's more important to focus on the most important details of each metabolic pathway. I want to detail for you what we think are the most important details that you need to understand to simplify metabolism so that you're focusing on the most important things that you need to know. The first most important detail that you need to understand are the inputs and the outputs. What is the starting material for each metabolic pathway? What are the products at the end of that pathway? And are there any cofactors along the way that are important? And last of all, how does this pathway fit into the bigger picture of metabolism? How does it fit in with the other pathways and how are they integrated together? The second important detail is to know the key enzymatic steps. Now, that doesn't mean that you need to learn each and every step. In fact, there's only a few steps in each pathway that are critical to understand. First of all, you need to know what is the rate-limiting step in the reaction. Which steps are regulated and what are they regulated by? Know the enzymes, the inputs, and the outputs for each of these steps. But the other ones aren't so important to understand. The third thing is to know something about diseases associated with each metabolic pathway. The USMLE is often organized around clinical vignettes and talks and asks questions about biochemistry in the context of human health and disease. So it's important to know which enzymatic steps have known genetic deficiencies causing disease. Which steps have other known dysfunctions that can lead to disease, including cofactor deficiencies? And what is the pathophysiology, the signs and symptoms, the inheritance, and the treatment for each of these diseases? If you can know just these important details about each of the metabolic pathways that we discuss, then you'll be well prepared to pass the biochemistry sections of Step 1 of the USMLE. Now I want to talk for a few minutes about the general concepts concerning the integrated control of metabolism. What are some of the general ways in which each metabolic pathway is controlled? Now there are really 2 types of regulation for each metabolic pathway. And we call these local regulation and hormonal regulation. Local regulation has to do with the control and coordination of metabolism of a single cell. And this is usually by mass action because of regulated by substrate concentrations, or by feedback regulation, that is regulation of a pathway by the product concentration. This occurs on a single cell level and has nothing to do with hormones or with the adjacent cells or other cells in the body. But this just deals with how metabolic pathways in a given single cell are regulated. But each of our cells makes up part of an entire organism. And so each of our cells need to be regulated in the context of the entire organism. And this coordinated regulation of metabolism is controlled by hormones. There are a lot of different hormones which can control metabolism. But for the purpose of the USMLE, it's really important that you understand just one, and that's the insulin-glucagon system. The primary concept in hormonal regulation is homeostasis. We want to maintain constancy of function in the face of a changing environment. For example, when we eat a meal and our blood glucose increases, we want to be able to bring that blood glucose back to a normal level. When we fast and our blood glucose drops, we need to have mechanisms to keep that blood glucose in the normal range, and so forth. Now, there are two antagonistic hormones called insulin and glucagon who signal the body to respond appropriately to maintain this homeostasis in both the fed and the fasted state. That is, the condition in which our blood glucose is high after a meal and the condition in which our blood glucose falls between meals. Glucagon is a polypeptide hormone that's produced by the alpha cells of the islets of Langerhans in the pancreas. Glucagon is secreted in response to falling blood glucose. That is, glucagon increases in the fasted state. Its function is to make available the storage forms of energy to use to raise blood glucose back to the normal level. Now, there are some other hormones that function similarly, and for the purposes of this review, we lump them together. And those are the catecholamines, like epinephrine and norepinephrine. The effect that they have on cellular metabolism is very similar to that of glucagon and can be considered together. Now, glucagon functions like most hormones by binding to a cell surface receptor. When it binds to its receptor, this receptor is activated, and the receptor activates what we call a heterotrimeric G protein. This heterotrimeric G protein then activates an enzyme that's bound to the membrane called adenylyl cyclase. Adenylyl cyclase takes a molecule of ATP and converts it to cyclic AMP. Cyclic AMP is what we call a second messenger. It's a connector between signaling pathways in the cell. Cyclic AMP activates a lot of things, but the most important one in terms of glucagon function is protein kinase A. Protein kinase A is a protein which phosphorylates other proteins, and it's a key regulator of the response to glucagon in a given cell. Protein kinase A phosphorylates a lot of enzymes, and those enzymes then either increase or decrease their activity in response to that phosphorylation. Now, whether they increase or decrease their activity depends on whether or not that pathway should be activated or inactivated in the fasted state. Now, as we go through metabolism, we'll give you a lot of examples of this particular type of regulation. Insulin, on the other hand, is also a polypeptide, but it's produced by the beta cells in the islets of Langerhans of the pancreas. Unlike glucagon, insulin is secreted in response to rising blood glucose, signaling a fed state. This is the hormone that goes up after you eat a meal. Its function is to transport glucose into cells and convert it to storage forms of energy like glycogen or fat, so that these storage forms of energy will be available in the fasted state. Insulin also binds to a cell surface receptor, but this cell surface receptor, rather than being associated with a heterotrimeric G protein, is associated with a different type of signaling system. In this case, what we call a receptor tyrosine kinase, or RTK. Binding of insulin causes dimerization of the insulin receptor, and the two receptors come together and phosphorylate each other at tyrosine residues. That's why we call it a receptor tyrosine kinase. There are effector proteins in the cell which bind these phosphotyrosines on the insulin receptor and transduce this signal into the cell. Insulin, like glucagon, has a lot of different effects, but we'll mainly consider just two. The first thing that happens when the insulin system is activated is that you get activation of protein phosphatases that dephosphorylate enzymes, especially those enzymes which are phosphorylated by protein kinase A. In addition to that, it also activates a cyclic AMP phosphodiesterase. This is an enzyme which cleaves cyclic AMP to its inactive form, 5' AMP. Now, you'll notice that both of these effects directly oppose those of glucagon. So, what I want you to really understand, the basic concept I want you to understand, is that the effect of insulin and the effect of glucagon is exactly opposite to each other. Glucagon increases when blood glucose falls. Insulin increases when blood glucose increases. Glucagon activates a heterotrimeric G protein, which activates adenylyl cyclase, which produces cyclic AMP, activates protein kinase A, and leads to the phosphorylation of a lot of enzymes. Insulin leads to phosphorylation of tyrosine amino acids on its receptor, which causes effector proteins to lead to protein dephosphorylation. So glucagon leads to phosphorylation, insulin leads to dephosphorylation. These two things are exactly opposite one another, and we'll see examples of that as we go along in considering the details of each individual metabolic pathway. Now, the last part of this section, I briefly want to introduce you to carbohydrates. We're going to be talking about carbohydrates a lot in subsequent lectures, and so there's some basic concepts that you need to understand about carbohydrates. Carbohydrates are molecules that have the general formula CnH2nOn. For example, a molecule of glucose is C6H12O6. The basic units of carbohydrates are called monosaccharides, and these are carbon rings of either 5 or 6 carbons. The 5 carbon rings are called pentoses. The 6 carbon rings are called hexoses. And a schematic diagram of one of these hexoses is shown at the right. You can see that they form rings, typically. The most common pentose is a molecule that we call ribose, and we'll encounter that in some of the metabolic pathways that we're going to discuss. Common hexoses include things like glucose, fructose, and galactose. Now, monosaccharide units are often linked to each other by an O-glycosidic bond. That means that there's an oxygen in common between these two molecules that binds them together in an ether-type linkage. The most common disaccharides are sucrose, which consists of a molecule of glucose and a molecule of fructose, lactose, which consists of a molecule of glucose and a molecule of galactose, and maltose, which consists of 2 glucose molecules bound to each other. Now, these 3 disaccharides can be found in many of the foods that we eat, and so it's important for us to understand how they're broken down so that they can be absorbed and utilized by the metabolic pathways in our body. The other form that carbohydrates can take is polysaccharides. Multiple monosaccharide units can be joined together by these same O-glycosidic bonds, these same ether linkages, to form polysaccharides. The most common polysaccharides are glycogen, which are long chains of glucose, which are found in animals. It's a storage form of carbohydrate that's found in animals, and thus in animal products that we eat. The other common polysaccharides are starches. These are the storage form of carbohydrates in plants. These are things like amylose and amylopectin. The last one is dextran. This is a polysaccharide that's found in bacteria and yeast, and so it's less common in the human diet. In order for us to utilize these carbohydrates in our diet, we have to digest them first. Our small intestine can only absorb monosaccharides, and so we have to break down polysaccharides into monosaccharides in order to absorb them. And this process starts in the mouth. There's an enzyme called alpha amylase, which is produced by salivary glands. It's also produced by the pancreas. And this breaks down starches, these long carbohydrates, into smaller bits. It breaks down starch to maltose, which is 2 glucose molecules bound together, or maltotriose, which is 3 glucose molecules bound together, and alpha-limit dextrans, which are disaccharides that have a different O-glycosidic bond. These disaccharides and oligosaccharides then are digested further in the small intestine. The products of starch digestion, this maltose and maltotriose and alpha-limit dextrans, are further digested to glucose by maltase and alpha-dextranase. The dietary disaccharides, things like sucrose and lactose, are broken down by enzymes called sucrase and lactates to their component monosaccharides. These monosaccharides then are absorbed by the intestinal epithelial cells, along with a molecule of sodium. Because this transport is against its concentration gradient, that is, the concentration of these monosaccharides inside the cell is higher than those outside the cell. So because it has to pump these molecules against the concentration gradient, an input of energy in the form of ATP is required, as depicted at the right. From the intestinal epithelial cells, these monosaccharides are transported via the portal vein to the liver. Once they reach the liver, then they have one of several fates. You can take these monosaccharides and oxidize them completely to carbon dioxide and water with the release of energy. You can store them as glycogen in the liver and muscle. You can convert them to fat in the liver and to form triglycerides, and these are stored in adipose tissue. Or you can release them into general circulation for metabolism by other tissues. So this ends our introductory section to metabolism.